Okay, so we're moving on to special senses. The last material, should be the last material. You see the autonomic nervous system and special senses are our last chapters before exam 3. That's my dog barking at somebody, so sorry about that. I'm at home doing this. All right, so special senses: vision, associated with sight; olfaction, smell; gustation, taste; remember, hearing and equilibrium. So everything about special senses is special, I keep saying. You know, the receptors, instead of being just nervous system structures that act as a receptor, these are cells that are actually going to act as a receptor. The sensory neurons are bipolar, not unipolars, and they have their own area on the cerebral cortex to be processed. Right, so vision, the receptors are located in the retina. We have accessory structures, the eyebrows and eyelids to help protect the eye. They also help capture like the eyebrow sweat from getting into the eye. The conjunctiva are mucous membranes on the eyelids. You know, there are two surfaces, one that's actually intimate with the eye itself, and then one portion of it is on the inferior surface of your eyelid. And they help prevent the eye from drying out. It also prevents material from rolling to the back of the eye. We have two portions of it, a palpable and ocular portion. The ocular portion is the part of that membrane that's on the eyeball itself, and the palpable portion is on the under surface of your eyelid. We have extrinsic muscles of the eye, skeletal muscles that move the eyeball. They are controlled by, remember, 3, 4, and 6 cranial nerves: ocular motor, trochlear, and abducens. And we have lacrimal glands that are located to the superlateral aspect of the eye, so the upper outer margins, closer to your temples, and they are dilute saline solution containing mucus, antibodies, and different antimicrobial or antibacterial lysozymes. Here we can see the lacrimal system. I love this because you really see essentially why your ears, nose, and throat are connected to one another. You have the lacrimal glands with lacrimal ducts that secrete tears to the undersurface of the eyelid onto the surface of the eyeball. They travel medially to what's called the medial canthus, which is the medial fold of your eyelid, so to speak. And these little openings, these black dots on the upper lid and the lower lid, are called the lacrimal punctum. And tears will flow, or the fluid will flow into that and travel along the canaliculi of the lacrimal system, another canaliculi, on the upper lid and the lower lid. And then they merge here in the lacrimal duct, or or like the nasal lacrimal, duct system, to the nose. Right, accessory muscles. What we have are extrinsic muscles of the eye. Right, so we have these 6 muscles. We have 4 rectus muscles and 2 oblique muscles. So the rectus muscles are found at, if you look at a clock face, 12, 6, 9, and 3, right, at those quadrant points. The oblique muscles run in oblique fashion, so at kind of a 45-degree angle. You have one on the superior hemisphere of the eye and one on the inferior hemisphere of the eye. So we have a superior and inferior rectus muscle, and then the oblique muscles, I should say, and then a superior, inferior, lateral, and medial rectus muscle. And these will become a lot more apparent when we just look at models of the eye, especially in lab. But here we have we're looking at the lateral aspect of the eye, right? The models in lab look just like this, are very similar to it. Here you have the nose, right? Anteriorly, there's your nasal bone, right? And you're looking into the eye here. So we have, to the lateral aspect, these fibers, the rectus muscle fibers, travel directly back to the posterior aspect of the eye, kind of like if you pull your hair into a ponytail. So those fibers, look at the direction of your muscle fibers that tell you the difference between a rectus muscle and an oblique muscle. So we have lateral rectus here. We have inferior rectus to the bottom, This one that's running looks kind of like a hammock. That's your inferior oblique muscle. To the top surface, traveling directly backward, is our superior rectus. And then we can see superior oblique. Now, under superior rectus, you see that white connective tissue? The tendon travels anteriorly and medially toward your nose, and then it passes through a little trochlea, and then you can see the muscle belly, the red portion of the superior oblique muscle. If we could see the other side of the eye, you would see the medial rectus muscle. If we look, anterior to posterior to an eye, then we can see all of the muscles: superior rectus, inferior rectus, medial rectus, lateral rectus, superior oblique to the top, passing through that little trochlea, and inferior oblique on the bottom. We can come back to this image and maybe label up the cranial nerves associated with it. I think that's a good study technique. Okay, so ocular motor, here are your 3, 4, and 6, your cranial nerves associated with eye movement. We said we were going to come back to that and start to put together, which muscles are innervated by which cranial nerve, and ocular motor innervates most of them. So ocular motor innervates superior, middle, and inferior rectus muscles and the inferior oblique. The abducens muscle innervates lateral rectus, and the trochlea, cranial nerve, innervates superior oblique. There are a couple different ways to remember this. If you set up a little chart, I always put, and I put the cranial nerve by number. So we have cranial nerve 3, and next to that as the nerve, and then we would have, let's start with abducens and trochlear, because each of those only innervates one muscle. But if you have LR for the muscle, and then 6, lateral rectus innervated by cranial nerve 6, SO for superior oblique, and next to that 4, and then all else, which are all the other muscles, 3 for ocular motor. So LR6, lateral rectus, cranial nerve 6, abducens, because when lateral rectus, the muscle to the outer side of your eye contracts, it laterally deviates the eye. So you look, if the right lateral rectus muscle is contracting, the right eye will move out laterally toward your ear, so you look to the side. So that's called abduction, so that's abducens nerve. The trochlear nerve innervates superior oblique. The superior oblique courses anteriorly and passes through the trochlea before it travels on the medial aspect of the orbit of the eye. So that is innervated by trochlear nerve. And then anything other than lateral rectus, LR6, SO4, are all innervated by ocular motor. Okay, homeostatic imbalances. We have conjunctivitis and pink eye, which are technically 2 different things, but often red eyes of conjunctivitis will accompany pink eye. Conjunctivitis is technically just inflammation of the conjunctiva, those membranes to the undersurface of the eyelid and on the surface of the eyeball. But it's often associated with infection, and that's pink eye. That's a conjunctival infection caused by bacteria or viruses, and it's highly contagious. So when someone has that, you really need to make sure, do the best you can, that one eye doesn't infect the other. Yeah. Have separate hand towels for those individuals. Carefully wash your face, with a hand towel, face cloth, and change that every day. Wash your first eye, that's uninfected, and then kind of go to an infected side. All right, so conjunctivitis, just inflammation. Pink eye, is an infection of bacteria or virus. Okay, eyeball structures. We have tunics. The eye is organized into 3 layers. You're going to find as we traverse and course through EMP2 that we have 3 layers, and 3 layers of things. So we have an outer layer called the fibrous tunic, the middle layer called the vascular tunic, and then deepest layer, the sensory or neural tunic. So most structures are going to be fine that you need to identify and know the functions that are found within those 3 layers. But we have other structures that are not considered part of a tunic, but they're there. Right? So we have the lens, We have the chambers of the eye, anterior and posteriorly, and then we have the optic nerve. All right, so this is a great picture for color coding and looking at where these structures are located and in which tunic or layer. So let's start with the outer white, right? The outer white portion. Its majority of that is called the sclera. That's the whites of your eyes. So that's all that white portion. And directly anteriorly over to the left-hand side, that's your cornea. Right? And that lets light pass through and then enter into the interior of the eye. So the most outer layer is called the fibrous tunic. The layer is called tunic is another term for layer in Latin. So the outer layer, fibrous tunic, is composed of the sclera and cornea. Then deep to that, you see that pinkish reddish layer. That is your vascular tunic, thus they indicate it in red, right? Because there's probably a lot of blood there. Well, there is. The majority of the eye, all the way from the optic nerve, which is leaving the right side of the eye, you can see all that yellow, the optic nerve there, leaving the kind of at about 4 o'clock or so. And that's your optic nerve. All right, so the red portion from there all the way coursing anteriorly, that is called the choroid. Then you see this kind of wider teardrop area here, that's called the ciliary body. And then further anteriorly is your iris. Okay. Then the deepest layer is the sensory or neural tunic. That's all the portions of it that is associated with the nervous system and vision, receptors for vision. That's that yellow layer. So that will include the retina, which is all in yellow, and the optic nerve leading in the back. All right, so the fibrous tunic is the most outer layer made of dense avascular connective tissue. There are two structures that are found there, the sclera, which are the whites of the eye, to help protect and give the eyeball shape. It's also a good surface for those muscles to attach to, the tendons of those extrinsic muscles of the eye. And then the cornea is a transparent anterior portion that allows light to enter. And because it's curved, it's going to refract light. Refract light means to bend light photons. It's going to kind of act along with other structures to funnel these light photons and direct them right to the surface of the retina at a particular area called the central fovea, or the fovea centralis. Right, that vascular tunic is the layer deep to that. It includes the choroid, that's highly vascularized, right, a pigmented portion of the posterior aspect of the eye, and the majority of that layer as well. Right, helps to absorb light photons and keep them from kind of reflecting and bouncing around in the eye. You have the ciliary body that contains ciliary muscles, which control the lens's shape. So that lens, we're going to look at it. We have these muscles that have what's called suspensory ligaments attached to the lens. And depending on whether we want to let that lens kind of puff up like you're blowing up a balloon or flatten like you're kind of deflating a balloon, will determine, how we want to be, again, bending these light photons, this light that's entering the eyeball so that we can focus it on the central fovea. It also contains cells that produce what's called aqueous humor, and aqueous humor is the fluid that's found in the anterior chamber of the eye. So anterior chamber, aqueous humor, A to A. The iris is the anterior portion, of what you consider the pigmented portion of your eye. That's what would determine, the surface of it, whether you have brown eyes, blue eyes, green eyes. And so we're going to look at that, 2 layers of muscles there. It helps regulate the amount of light entering the eye by controlling the size of the pupil, pupillary constrictor, pupillary dilate, like we saw in the autonomic nervous system. And the pupil is just a hole in the iris where light can pass through. All right, so the iris has 2 layers of muscles. We have the sphincter, what's called the sphincter pupillae, or the inner circular muscles. And when they're stimulated, they pupillary constrict and they close down the pupil, so that less light passes through it. Which branch of the autonomic nervous system would stimulate this reaction? That's the parasympathetic division. Then we have, lateral to that, so to speak, we have the dilator pupillae, or the outer radial muscles. And upon contraction, they stimulate pupillary dilation, so they make the pupil larger in diameter. What branch of the autonomic nervous system? Sympathetic. So here we can see the picture, the circular muscles to the interior and the radial muscles, kind of lateral, superficial to that, so to speak. And so it says parasympathetic division stimulates those circular muscles to constrict, and it causes pupillary constriction. The sympathetic division, see, it stimulates the radial muscles to contract and pulls open the pupil, and thus pupillary dilates. Right, the sensory tunic, the retina is composed of 3 layers of cells of the retina. You have the photoreceptor cells, the bipolar cells, and the ganglion cells, and we'll look at these on a slide as well histologically. The photoreceptor cells, which are closest to the choroid, that's the best way to describe it. This is where what's called transduction of light to greater potentials occur. So we have these cells that are going to act as receptors, right? Photoreceptors, because they respond to light photons. And we have rods that respond to dim light and cones that respond to bright light and color vision. These are found in the highest concentration in that area, of visual acuity, that we're going to look at. That's where we want to funnel all these light photons to this area of visual acuity where we have lots of cones. Then we have the bipolar cells, right, that receive the greater potentials that are generated from those receptor cells. And that then generates an action potential on the 3rd layer of cells called the ganglion cells. And the ganglion cells ultimately form the optic nerve and lead the back of the eye. Then that information becomes processed on the occipital lobe. And so we're going to look at that. We have the optic disc, is where the optic nerve exits the eye. That is what we consider the blind spot, because it will be devoid of any photoreceptors. We not only have the nerve exiting and entering, or exiting, I should say, it's only going one direction, but blood vessels. We have arteries entering the eye and veins transporting blood away from the eye there as well. And then we have another little diffuse area called the central fovea, or the fovea centralis. This is our area of visual acuity. We have lots and lots of cones only. All right, so here in the upper left-hand corner, so okay, let's take a little box and let's look over to the right of it. Is, what do we find there? Well, we can see the yellow layer, that's our sensory or neural tunic. Right, those are going to be, the nerves from your ganglion cells that ultimately form the optic nerve leaving it. And you can see those red and blue vessels here. All right, then below that, you see where we can see, sort of, even without magnifying this, 3 layers of cells, and that is the retina. Retina contains 3 layers of cells. So now over to the bottom left-hand side, we go, okay, the yellow portion there over on the left-hand side, those are the axons of the ganglion cells, and the ganglion cells are those kind of paler staining first layers, so to speak, that you can see, on the slide. And they're closest to the lumen of the eye, where we have what's called vitreous humor. All right, so we have the ganglion cells. Then we have the pink layer of cells called the bipolar cells. And then farther to the right on that same image, we have the photoreceptor cells. Over to the right, that's a really good picture of a histological slide of what the retina will look like. And they look very similar to that. Not quite as perfect as that in lab. So we have the retina, the whole layer there, those 3 layers of cells is the retina. We have the ganglion cells, then we have the bipolar cells, and then we have the photoreceptor cells. That is the retina. 3 layers of cells, always in, that same sequence, so to speak. I find that one of the easiest ways to get orientation is to use the choroid, which is over to the right-hand side. The choroid is highly vascularized, and it looks like dried blood, doesn't it? It's all this red, blackish, brownish kind of material. So that is part of the vascular tunic. Anything to the left of it, when you find the retina, then next to the choroid, you will have the photoreceptor cells, and then a bipolar and a ganglion cell in that order. Whether it's going left to right or right to left, it doesn't, just depends on the way the slide is oriented. The lens is a biconvex structure that can change shape to focus light on the retina. This is the one that's going to be acted upon by the muscles of the ciliary body. The lens also divides the eye into an anterior and posterior chamber, or sometimes referred to as a segment. So from the lens to behind the cornea is the anterior chamber, and that's filled with aqueous humor. That fluid gets replenished and pumped and circulates to the anterior aspect of the eye, bringing a lot of nutrients, oxygen and nutrients to those tissues. So it's replenished through life. But from the lens to the back of the eye to the retina and to the optic nerve is the posterior chamber, and that contains vitreous humor. That fluid does not get replenished. It's more gelatinous, it's thicker, like a higher density, and it keeps the retina kind of pushed against, the walls to kind of keep connected to the choroid. So this is how your aqueous humor is produced. Okay, so over on the left, it's saying, okay, let's take a look at that lens. Right, here's that biconvex structure. All right, and the fluid, right, that aqueous humor is produced by the ciliary, some cells here in the ciliary body. Right? It travels to the anterior aspect of the lens through the pupillary opening, and then nourishes the whole back of the cornea. And then it gets absorbed into this little canal showing here in blue, called the Canal of Schlemm. I think they have changed it now to the sclerovenous something. I don't know. I think it's easy to remember the Canal of Schlemm. I'm not going to ask you about it, but just for people who want to know, right, is that fluid is produced, circulates to the anterior aspect and chamber of the eye, and then gets sucked up into that other canal, Schlemm. Sort of like cerebrospinal fluid. We pump it out, and then it gets kind of filtered and absorbed, around in the skull. So this fluid has to have a place to go. If that canal gets blocked, it produces, ultimately, it may produce glaucoma. All right, so our homeostatic imbalance is glaucoma, a condition in which the drainage of aqueous humor is blocked, causing fluid to back up and increase the pressure in the anterior aspect of the eye. Pressure may increase to dangerous levels and compress the retina and the optic nerve, leading to blindness. Symptoms, well, there are very few early symptoms, but late symptoms or signs include seeing halos around lights and blurred vision. So let me just back up here and let me, oopsie doodle, go back here. So, when we look at the overall scheme of the eye in the upper left-hand corner here, what we're saying is that if that fluid does not drain into that little canal, right, that fluid pushes back on the lens, which pushes back on the vitreous humor in the posterior chamber, which puts pressure on the nerve leading the posterior aspect of the eye. And that's not good. Okay, your visual pathway. Well, it's through the eye. If we look at the direction that light photons will travel, if you just start in the front of the eye and you go structure all the way to passing through the retina, then this will make sense. So the first structure that light photons will pass through is the cornea to the front, where you might put a contact lens. And the cornea refracts or bends most of those light photons, so it starts to funnel it through the aqueous humor, which is the fluid to the anterior aspect of the eye, through the pupil to the lens. The lens also can bend those light photons depending on how close the object is or how far away it is, in that the ciliary body will help flatten or let that lens puff up depending on where we need to direct, the light photons. It will pass through the fluid in the posterior chamber called the vitreous humor to the retina to ultimately the photoreceptors. So through the retina, light changes chemicals in the photopigments in the rods and the cones, those are the photoreceptors, which generate the greater potentials in a ganglion in the bipolar cell. The bipolar cell creates a action potential on the ganglion cells, and that action potential travels on the ganglion cell to the optic nerve. Those impulses travel from each optic nerve on the back of the eye to where it crosses, called the optic chiasm, and then travels through the thalamus to be directed to the appropriate area on the occipital lobe. So light photons essentially will pass through the ganglion cells, the bipolar cells, to the photoreceptor cells. And then from there, those photoreceptor cells will ultimately create graded potentials on our bipolars, which produce action potentials on the surface of those ganglion cells and down the axons to the, down their axons, which form the optic nerves and ultimately get processed on the occipital lobe. That's your primary visual cortex on the occipital lobe. All right, more homeostatic imbalances. Well, problems associated with the diffraction of light related to the eyeball shape. And this is not uncommon for most people. You can either have one of these or a combination of all three, which now at my age I have. We have myopia, hyperopia, and astigmatism. These are just a few. These are the main ones. Myopia means nearsightedness, which means you can see things close, but not far away. So what happens is the eyeball is of a different shape. It's too long so that the focal point, which is where all these photons will essentially merge, is in front of the retina. Hyperopia means you're farsighted. You can see things far away, but not close up. This will be the opposite. The eyeball is too short so that the point, your focal point is behind the retina, where these light photons essentially cross. It's going to be behind the retina. I'll show you a picture in just a minute. Astigmatism is an unequal curvature in parts of the cornea or the lens, and therefore they don't direct the light photons appropriately. There are tests that you can do for that, and there's adaptations to the lens, either in your glasses or even contact lenses now that will address that. So I love this picture because it lets you see essentially what we're talking about here. So over to the left-hand side, the emmetropic eye means it's a normal eye. So as light photons pass through, and those are those kind of reddish lines, it's going to pass through the cornea and through the lens, and you see how they cause these light photons to funnel down and merge so that they land right on the surface of your central fovea, of your fovea centralis, your area of visual acuity. Right. Below that, what if we have an eyeball that's more egg-shaped and longer than a normal eye? Right, that's called myopicism. So you see how the cornea and the lens, they are bending the light photons correctly, but you see that point where they cross, and then they actually start to diverge beyond that. Right. They are merging and crossing. That focal point is anterior in the vitreous humor of the eye. And thus the image is actually trying to be formed to the front of the eye. A hyperopic eye, or farsightedness, the eyeball is a little shorter than the normal, and that point of conversion is behind the eye. So over to the right, it shows you the shape of the different lenses and how you're going to correct and bend those light photons before they enter the cornea and lens so that they ultimately, boom, merge and intersect on the central fovea of both of those eyes. So for people who wear glasses and things, I think that's interesting to see. Color blindness, another homeostatic imbalance, means we lack one or more of cone pigments. It's either red, blue, or green, or it could be more than one. They are inherited, they're carried on the X chromosome. So it's going to be more common in males and females, way more common. Males, their genetic makeup to determine gender is one X and one Y, and in females, there are two X's. We'll look at how that's inherited in AMP2. Okay, we're moving on to chemical senses, and the chemical senses or chemoreceptors are part of smell and taste, olfaction and gustation. Right, so smell and taste, or olfaction and gustation, right, they're complementary senses to let us know whether a substance should be savored or avoided. Chemoreceptors are used by these systems, so we're going to have cells that act as chemoreceptors. Chemicals must be dissolved in an aqueous solution, a water base, to be picked up by the chemoreceptor. Smell receptors are excited by chemicals dissolved in nasal fluids. Taste receptors respond to chemicals dissolved in saliva. Just want you to tell you that those particular, and I don't know why it's not here. I think farther down, and we'll talk about this. All right. It had to do with a term for that, tastings and things. I'm sure it's down in the notes someplace. All right, so your sense of smell. Let's start with smell. We have the olfactory epithelium. Remember that cranial nerve 1 is the olfactory nerve, and we have those olfactory foramina in the ethmoid bone for these nerves to pass through and line your nasal cavities. So they're located in the roof of your nasal cavities and quite, a large area. They contain olfactory sensory neurons. Those are bipolar neurons that will convey, the stimulus of smell. Olfactory neurons are covered by mucus. Thus, mucus is very watery. That's the watery, that's the aqueous solution for these different chemicals to be dissolved in for those receptors to detect their presence. And this is what I was looking for. So covered on the solvent for odorants. So odorants are the chemicals that will be dissolved that can stimulate those different receptors or olfactory receptors. Olfactory neurons, unlike other neurons, have stem cells that give rise to new neurons every 30 to 60 days. So that's kind of helpful. Right, so here we have over to the left, we see kind of a half head where we can see the nasal cavities here and those different nasals called nasal conchae. And we can see how the nerves line that area. Over to the right-hand side, it gives you a better idea, because it magnifies it, but it's showing where those nerves are passing through the olfactory foramina of the ethmoid bone. And so, fracture or dislocation, so to speak, of the ethmoid bone can cut those nerves and lose a significant amount, if not all, of your sense of smell. Right, so the olfactory pathway, olfactory receptors extend through the olfactory foramina in the ethmoid bone to the nasal cavity. Into the nasal cavity, olfactory nerves carry impulses to the olfactory bulb, to the olfactory tracts. The olfactory bulb, don't be too concerned about it. It's really what we're using to identify the olfactory nerve when we're looking at the undersurface, of the brain. And you can see where that little bulb area, it's just to the superior aspect of the ethmoid bone, superior to the olfactory foramina. And then from there, the impulse are going to travel on these olfactory tracts to the olfactory area in the temporal lobe. And remember that it shares a significant amount of that information with the limbic system, so we have a lot of emotion and memory associated with smell. Gustation and taste, the repeating structural unit, so to speak, of taste is a taste bud. And this taste bud, we're going to look at that, contains gustatory cells that act as receptors. Most taste receptors are located in the oral cavity, in the papilla, on the tongue. Those are the kind of the indentations. When we looked at the cerebrum when we had gyri and sulci, this is what the tongue surface is going to look like. It's stratified squamous, like on the surface of your body and your skin. But again, because some of that tissue kind of gets compressed in there, it's like the cerebral cortex. You will have bumps and troughs to try to create room for all that tissue. And we will see then how these taste buds open up into that indentation. It's kind of like a sulcus. Basic taste sensations: sweet, sour, salty, bitter, and umami. That's considered to be a savory or meaty sensation. The lowest threshold is bitter. Right, so they're saying that, of those receptors, the one that's going to be stimulated the easiest and the fastest will be bitter. And that's because most toxins or things that can be harmful to you are generally bitter. They adapt very quickly in about 1 to 5 seconds. And tastants, unlike odorants, are associated with smell. Tastants are the molecules when they get dissolved in the aqueous solution of saliva, and contact a taste bud to generate an itch potential. Tastants versus odorants. Odor, smell, I think that's taste, tastants. Right, so here we're looking at the tongue. Different regions, have higher population of receptors associated with sweet, sour, salty, bitter, and umami. In the middle, where they show the surface of the tongue, you can see these different humps and troughs, humps and troughs. And you can see that indentation there. The taste buds would open to the inside surface of that papillary trough. And they're found on both sides. They're not just found on one side. All right, to the upper right-hand corner, that is one of those little purple little, kind of look like little sperms there almost. But if we magnify that, that is a taste bud. And that taste bud there is composed of those darker cells and those lighter staining cells. So we have basal cells that give rise to all these cells. They're sort of a stem cell. And then we have our gustatory cells. Over to the right, you can see how they're going to open up to a little pore area so that as food travels on the surface of your stratified squamous epithelium, because they're embedded in essentially an epidermal layer, is that the food dissolved, or those tastings dissolved in the aqueous solution of your saliva, can pass into the pore and then make contact with those gustatory cells that are receptors to the contents of the food, to the tastings. They will generate action potentials ultimately on those nerve fibers to the left-hand side. Below that shows you an actual slide of what it might look like. All right, so the gustatory pathway. Remember, gustation or taste is conveyed on 3 cranial nerves, 7, 9, and 10. So gustatory receptors are found in taste buds on 3, and conveyed, I should say, on 3 cranial nerves. To the anterior aspect of the tongue, or the 2/3, anterior 2/3 of the tongue, those sensations are carried on facial nerve. Posterior 1/3 of the tongue, glossopharyngeal, and the lower pharynx and epiglottis on vagus. The impulse travels to the thalamus and then ultimately directed, because the thalamus is that area that sorts and directs impulses, to the right, correct area of the cerebrum to be processed. So it's going to pass through the thalamus to the gustatory center in the insular lobe. In lab, they might refer to it as put it on the parietal lobe. That's fine. Loss of 80% of taste, or you lose 80% of the sensation of taste if the olfactory receptors are blocked or damaged. So they don't have to be damaged and cut to lose the sensation, of taste. So if we block the olfactory receptors, let's say you're congested because you have a cold, then you're not going to be as sensitive to the different odors or smells, especially if you're eating food or you're cooking. And olfaction is a huge part of taste, believe it or not. Right? And so it actually is about 80% of what contributes to how effective you perceive and process taste stimuli. All right, we're going to move on to hearing and equilibrium because we're getting in the ears. All right, so now we have to look at the ear anatomy. We have the outer ear, composed of the pinna, and the external auditory canal or meatus in your tympanic membrane. So we're essentially dividing the ear into 3 regions: the outer ear, the middle ear, and the inner ear. And we're going to look at structure. Those 3 are composed, are considered part of the outer ear, the middle ear, and the inner ear. The middle ear is an air-filled chamber inside the temporal bone that contains the ossicles, which are these little bones, the eustachian tube, the oval membrane. And then you have the inner ear, that's the fluid-filled chamber that contains the cochlea, which is our organ for hearing, and the semicircular canals, that is associated with balance and equilibrium. All right, so starting with the outer ear, and we're going to look at a picture in just a moment. We have the pinna. That's the outer elastic cartilage structure that directs sound waves into the external auditory canal. Your external auditory canal or acoustic meatus directs sound waves to the tympanic membrane. So this is acting as a funnel. It also contains cerumen or earwax to trap foreign substances. The tympanic membrane is where sound waves will be converted to mechanical energy. So when the sound waves hit this, it puts this membrane or trampoline into motion and causes it to vibrate. Converts sound waves to mechanical energy. All right, so here we're looking at an ear. We have over to the left-hand side, that's your auricle or pinna. direct sound waves into the external acoustic or auditory meatus. And then ultimately, those sound waves hit the tympanic membrane, and that converts these sound waves into mechanical energy. So including the tympanic membrane, external acoustic meatus, or auditory canal, and the pinna, those are the structures of the external ear. The middle ear will be everything on the other side of the tympanic membrane. Right. So the middle ear contains the ear ossicles. They transmit vibrations that are generated from the tympanic membrane to what's called the oval window. The first bone is the malleus, that's attached to the eardrum. Then we have the incus, which is the middle bone, and the stapes that's going to attach to the oval window, another type of flexible membrane, like a trampoline. Also in that middle ear region is the eustachian tube, or the pharyngotympanic tube, that links the upper part of your throat, what's called the nasopharynx, to the middle ear. And that helps equalize air pressure on both sides of the tympanic membrane. That's what happens, when your ears pop, you're equilibrating the air pressure on both sides of your tympanic membrane. And then we also have in there oval and round windows. So we'll look at what those look like in a minute. They're all found in the middle ear. All right, so tympanic membrane, outer ear. We have the malleus, incus, stapes, in that order from lateral to medial, miss. And on the other side of the stapes will be another flexible membrane there. And so once those bones vibrate, it also puts that other membrane into vibration. And then everything on the other side in the inner ear, those fluids will start to vacillate and produce essentially like waves in them. Below that region, we have the round window. And then further inferiorly, we have the pharyngotympanic tube, or the eustachian tube. The inner ear contains both the semicircular canals and the cochlea. Those structures are fluid-filled chambers, and they have there are cells that have hairs that extend into the lumen where the fluid is moving. Sorry, my phone was ringing. Let me just put that on do not disturb. Whenever we have something physically moving, that's bending something or compressing something, those are, again, the mechanoreceptors. All right, so we're going to find mechanoreceptors here because we're going to have fluid moving and they're going to have little hairs that get bent. Now, the vestibule area is this little egg-shaped bony cavity that connects the semicircular canals to the cochlea, and that contains receptors associated with static equilibrium. We have static equilibrium and dynamic equilibrium. And so this little egg-shaped region, the vestibule. The semicircular canals are 3 fluid-filled hoops that have fluid within them. And when you move, this is what we call dynamic equilibrium. When you move in a linear, either forward, backward, side to side, up or down, or rotation, like you're turning around or you're turning your car around, let's say, rotational movement, it will send information about the direction that you're going to your brain. Dynamic equilibrium. The cochlea is kind of a snail shell, kind of an organ associated with hearing. And so that cochlea contains the receptor organ for hearing called the organ of Corti, and it ends at the round window. So we're going to look at that. Here we have the internal portion of the ear. So up in the upper left-hand side, they're going, okay, let's look on the inside, the inner ear region. All right, so let's start with the stapes down below, because that's part of the middle ear. We said if we peeled that back, we could see an oval window. And then just to the right-hand side, that's going to be the round window. That's the end of the cochlea, which is to the right-hand side. All right, so let's look at the vestibule, that little blue egg-shaped area there. That's your vestibule. Static equilibrium. And then we have our 3 fluid-filled hoops here, the semicircular canals, dynamic equilibrium. Mechanoreceptors found there. The nerve transporting information from that region is the vestibular portion of the vestibulocochlear nerve. Okay, Then we have the cochlea. It looks like a snail shell. Its nerve is the cochlear portion of the vestibulocochlear nerve. The cochlea, it contains your organs for hearing, and it ends at the round finger. So I think what's helpful to try to understand the organization of the cochlea is if we picture it like, have you ever made a jelly roll or cooked something like that, baked a jelly roll? You have a flat sheet of cake and you put jelly in the center and then you roll it up and then you kind of cut it, lengthwise, in cross section, so to speak. So when a piece falls over, you can see the spiral effect in the interior. Well, that's what the cochlea is like. So if you could try to unroll that cochlea, that snail shell, so it's flat, you would see these 3 fluid-filled chambers. So here up on the top, that's what it would look like. Here's your cochlea. And if we unrolled it, we would find that one section has these 3 different vestibuli. You have the scala vestibuli, the scala media, and let me just say that there's scalas, they're not all vestibuli, I misspoke. Scala vestibuli, scala media, or what's now called the cochlear duct, and the scala tympani. 3 regions, 3 different tube systems. Well, I take that back. The scala vestibuli will actually become the scala tympani, but they have a certain type of fluid found there, and then another fluid found in the cochlear duct. If we further unroll this over to the right-hand side at 3 o'clock, this is what we refer to as this organ of hearing. We have a tectorial membrane to the top that's inflexible and bony, and then below that we have these supporting cells, and we're going to look at that anatomy in just a minute. Okay, so your auditory pathway, sound enters, it gets funneled by the pinna into the external auditory meatus or canal. Sound waves strike the tympanic membrane and cause it to vibrate. The vibrations are transferred through the malleus incus stapes, the ear ossicles, to the oval window, and then to the other side of it, to the vestibule, to the inner ear. These vibrations create fluid waves in the cochlea. It's going to travel on the scala vestibuli to the scala tympani and ultimately cause the fluid in the cochlear duct to move. It stimulates the hair cells, which are the receptors of the organ of Corti, to hit that tectorial membrane and bend the hair cells there. That generates an action potential in the cochlear nerve, a branch of the vestibulocochlear nerve. And that impulse then travels to the membrane, to the thalamus, and to the auditory cortex, the primary auditory area on the temporal lobe. This picture, I think, sums it up, at least, structure to structure, when you go from the outer ear to the middle ear to the inner ear, because that cochlea essentially got, stretched out and unrolled. So the impulses, or the sound waves, are traveling at 1 to hit the tympanic membrane, then malleus incus stapes vibrate, the oval window vibrates, and then it puts the fluid in the first portion, the scala vestibuli, into motion. Right, at number 3, it travels down that tube to where it makes a hairpin turn at what's called the helicotrema. And now that continues that tube. Now we call it the scala tympani. So it's essentially the same hose. It's just after this hairpin turn portion called the helicotrema, we call it the scala tympani. And that fluid gets put into motion until it reaches the round window, which acts kind of as a shock absorber, so to speak. Right. What happens in this cochlear duct, or the scala media, which is between those two, right, is where we find the hair cells of the organ of Corti. You'll see that more in lab. Let me go back and let me just point to it again. All right, so here, scala vestibuli to the top, scala tympani to the bottom, scala media of the cochlear duct in between. So when the fluid in the scala vestibuli starts to have these waves, that gets translated to waves down in the scala tympani below it. And both of those cause the fluid in the cochlear duct to have waves, essentially. All right, so what happens over to the right-hand side? Those hair cells of the organ of Corti hit that tectorial membrane to the top. That is a fixed roof. And when the bottom part of it starts to bob up and down, those hairs to the top part, says the outer hairs of those cells, hit that tectorial membrane and they bend, and that generates graded potentials and then action potentials on the cochlear nerve. Let me just see, make sure that they. Okay. Okay, and this kind of shows you the pathway. So here down below, we have Scala vestibuli, Scala Tympani, puts the prayer cells of the organ of Corti into motion. They generate graded potentials and then action potentials on the cochlear nerve, and it gets transported to the thalamus and then directed to the area associated with hearing on the temporal lobe. So, okay, we did that. All right, equilibrium. So this is the input from those semicircular canals. Those, as fluid moves, impulses from the vestibule or the semicircular canals will be carried on the vestibular branch of the vestibulocochlear nerve. Right? The brainstem, it shares it with the brainstem, helps activate reflexes in the body and extrinsic muscles of the eye. Remember the midbrain. And the cerebellum, to coordinate skeletal muscles to maintain balance. Right, deafness. We have two types of deafness, and it's really associated with the region that you're having the problem with. You can either have conduction deafness or general categories of conduction deafness versus sensory neural deafness. Conduction deafness is the result of damage to structures which conduct sound waves, the tympanic membrane, the generation of the ear ossicles, essentially everything found, either in the outer or middle ear. If we have sensory neural deafness, that is a result of the neural conductors, like the hair cells, the cochlea or the cochlear nerve. So essentially anything to the internal ear, any of those structures, all the way till it's processed on the temporal lobe. Right? That's considered a sensory neural deafness. So anything associated with a structure not functioning correctly that's found in the external or middle ear, conduction deafness. Sensory neural deafness would involve some structure anywhere along the pathway in where it's generated in the internal ear and the cochlea, and the nerve that transports it to the cerebral cortex area that processes it on the temporal lobe. And that is the end of special senses.